Okay, I lied. That wasn't a few seconds. That was less than 2. All right, so I mentioned there would be a list. Here it is. Fungi, bacteria, and viruses are the 3 people think of the most. But there are also these things called phytoplasmas that you pretty much only hear about if you're a plant pathogen or you're a plant pathogen geek kind of a person, ag geek. But they are these mycoplasma-like organisms. You can look them up. I'm not going to get into it, but they're not technically in the other categories. Another category at the bottom, nematodes, that's, I would say, the next on the list for most people. You've got your fungi, your bacteria, your viruses. Those are what people think of as pathogens. Nematodes are the only animal on the list. They are a microscopic roundworm. And because they are microscopic and attack and enter the plant and cause these pathogenic responses, disease responses, we do refer to them as pathogens. Unlike something larger where an insect is chewing on something, can that cause some damage? And can it even cause some pathogenic responses? Yeah. You have caterpillars that cause leaf rolling, but we don't call those pathogens. There's something about nematodes being microscopic. We look at them differently. At least pathogenic nematodes are microscopic. There's also algae and higher plants. Higher plants, the 2 that I bring up most often are dodder, which is D-O-D-D-E-R. And so D-O-D-D-E-R, if I go to images, it looks like somebody sprayed silly string. If you've ever had a can of the spray string, it's not nearly as popular as it used to be many years ago, but it's still around. But it looks like somebody sprayed some string all over your crop. And if you look really closely, it's wrapped around your crop plant and it has these attachments. See. So yeah, you can see how it's wrapped around. Usually when I've done these searches, I get a really good picture of an attachment. And I'm not seeing a good one. Just, oh, here it is. Or at least here's one. You can see these almost like octopus tentacle pads. And those things attach to the plant and will feed on it. It doesn't have any of its own photosynthetic capabilities. And so it's a parasite. Causes a lot of plant damage and disease response. So the higher plants, these parasitic plants, are the only non-microscopic things that I'm aware of that are considered pathogens. And the other one, mistletoe. And mistletoe, you always get these red berries because of Christmas. They are not red berries. This is what mistletoe really looks like, with white poisonous berries. And this is what it looks like up in an oak or other compatible tree. And again, the red berries are fake, or they're taken from a holly bush to just make it look Christmassy. The green and red color is Christmassy, but the true berries on mistletoe are white, and they are deadly poisonous. You got to be careful. Not as poisonous as some things. You can handle them, but you definitely don't want to eat them, or kids to eat them for sure. Birds can eat them, and that's how the seeds spread. So this is parasitic. It does have its own chlorophyll, and it can feed itself some, but it also attacks the branch, drills down into the vascular tissue and steals the phloem nutrients, steals the sugar, and lives happily as a parasite. And I always wondered where this tradition came from and if anybody had a sick sense of humor when all of this started. But now, you know, stand under the mistletoe and give your love a kiss during the holidays. You know, you want to catch somebody under the mistletoe or not catch somebody under the mistletoe, depending on what the situation is. But we've got this symbol of love for the holidays, mistletoe. And what is it? It is a highly poisonous parasite. Somebody had a sense of humor with that one. Anyway, those are higher plants that are considered pathogenic. And here's just some examples. Here's a microscopic view of some nematodes. Here's an electron microscopic view of some viral particles. Here's a fungus that is with the reproductive structures, and a couple of types of bacteria on growth plates. So, laboratory techniques. You need to learn how to work with microorganisms, be able to work in sterile conditions. And so there's a process called sterile procedure. And if you're going to learn basic concepts of pathology, dealing especially with the viruses, bacteria, and fungi, you need to learn sterile technique. And that's a fundamental concept in pathology. Now, the level of sterility that's required depends on the organism you're dealing with. Sometimes sterile technique has to be extremely strictly adhered to, and other times you just have to do some stuff with much looser concerns. It's the same idea if you go to the doctor and you've got the flu. Is the doctor wearing a hazmat suit when you show up with the flu? No. The doctor's not wearing anything. Coronavirus, the doctor's wearing a mask. Ebola, the doctor is in a full hazmat suit. It's the same thing with plant pathogens. And for the most part, you're not trying to protect yourself. There are a couple of plant pathogens that can be harmful to humans, but the vast majority, you have to eat a bunch of it to be harmful. And in those cases, you know, that's a different situation. We're trying to protect the integrity of the experiment and the tests. We don't want to contaminate things because we're doing science. And if we contaminate it by poor procedures, we don't get good science data out of it. So what's the procedure? Pretty simple. You sterilize the surface of your plant materials and any of your tools. You excise or cut out a piece of the tissue that appears to be diseased or infected with some pathogen. You place that tissue that you've excised into some growth media. You try to allow some time for that pathogen to recover and start growing, and identify it if you can. So let's look at the procedures. Now, this is a very casual procedure. It's being done out in the open on a stainless steel bench. Stainless steel is good because it's easily sterilized and it doesn't carry contamination well at all. So it's an easy, sanitized, clean surface. But they're also not wearing gloves. You would most likely be doing this in what's called a laminar flow hood, which is a sterile hood, and wearing gloves if it was something where you were worried about contamination. For some pathology experiments, gloves is overkill. You don't need it, but anyway. So they clean the surface, they take the material that appears to be infected with some sort of possible pathogen. You take a diluted solution of some sort of cleaning agent, in this case just bleach is fine, and dip it in bleach of a certain concentration for a certain length of time. And then you clean your tools. This is alcohol in this container here, and they will dip their scalpel and tweezers into the alcohol and then put the alcohol coated stainless steel tool into a flame. And that the alcohol, if it's high enough concentration, 90% or more concentration, it'll easily burn and kill anything that might be on the stainless steel surface of your tools. So you call that flaming your tools. And it makes sure that you are being sterile with the next step, which is excising the material. You do have to be careful. You know, I've always set up my station where the alcohol is off further to the side and a little further out, so I'd have to reach for it and nothing goes near it except when I dip into it, because I don't want to grab something, accidentally tip over the alcohol, and boom, I've got now a fire, and it can spread. A splashing alcohol is really dangerous, so you want to be careful. You're dealing with a dangerous situation. So here they're excising a little piece. You can see they cut that piece out. And they're going to put it onto a plate which contains nutrients and agar. Agar is a gelatin-like material, just like Jello without the sugar and the food coloring. Same concept, but it just has special nutrients and special ingredients to encourage the growth of what you think the pathogen is. And you might have 3 or 4 plates that you're going to put it on because you don't know. So you're going to excise several pieces and put it on different plates. And maybe even put it in different temperatures, because maybe you don't know what the pathogen is and you've got to mess around till you find the right combination that will make the pathogen happy to grow again so you can identify it. And after you allow it to grow for a while, here's the plate. And so there's some morphological or physical structure that you can see here. It almost looks like tree rings. And the black color, the decreasing darkness, this alone, just looking with the naked eye, gives you some indication of what you're looking at. And then the researcher, here's Dr. Kawiki, is going to look under a microscope, look at the detail at the microscopic level for further data about what this pathogen is. And you might need to do some genetic testing or serology testing, like ELISA testing or something, to figure out what's going on. And here are just some diseases, root diseases. I believe, well, I'm not even going to go there. I'm not 100% sure. This is a viral disease of cilantro, perhaps. So we need to do a diagnosis, and that's really important, because if I don't know the diagnosis, I don't know what the pathogenic organism is, I can't tell you what to do about it. Your response to a disease is only going to be effective if you know what the pathogen is that's causing it, and possibly some of the related stresses. So there might be a pathogen, but there might be other stresses you can deal with as well. That's a little more complicated, but you're looking for those things. Is there also a nutritional deficiency? Is there also a heat problem or a soil problem or a moisture problem that could be contributing to the susceptibility to the pathogen you found? But you have to know what the pathogen is. You do the science of identifying the pathogen or diagnosing the pathogen based on the disease triangle. And so you look at, well, what is my host? If it's susceptible, it's got the disease, that's a clue. I look at the environment in which the disease is occurring. Again, more clues. And then you try to look at the pathogen itself. And so you're systematically doing science on the work. So you're going to collect information, interviewing the grower, anybody who has information about what happened, what you saw, how you fertilized, how you watered, did you sprinkle or irrigate at any point, you know, all of the things. You get information. Then you go out and you look at the site itself, gather some samples from the plants on your own. and you go inside, you look at the plant tissue immediately without doing the regrowing. You just look at things under the microscope. You're trying to gather information about what you think is going on. And then you can incubate the tissue in the chambers. And how you incubate them depends. I showed you a way to excise tissue and grow it up on plates, but there are different procedures for different kinds of organisms. There are commercial test kits out there, and I'll show you an example where they already are done. You can test for certain diseases right there at the tailgate of your truck. You can isolate and culture the pathogens, and that's the excise and grow material in a laboratory. And then you identify which isolate it is. If I know that I have downy mildew, well, downy mildew just tells me what genus and species I have. It doesn't tell me which isolate or which version it is. So for example, if I have lettuce, that's a genus and species, Lactuca sativa, but I don't know if I have butterhead lettuce, romaine lettuce, crisp head lettuce, and the same thing is true of pathogens. They have a genus and species, but there are lots of different types, just like humans. There's lots of variation, and you need to sometimes know which pathotype or isolate you have in order to know what to do about it. And then in parentheses, we have demonstrate pathogenicity. You may or may not do that if you're doing a research paper, you're doing university research. You need to close the circle. You need to go all the way around. So if I'm given a plant that has some symptoms of disease and I isolate a pathogen and grow it up, just because I grew something up doesn't mean I found the right thing. There's probably dozens of fungal spores on every sample that go into the lab for testing. Did you grow the right one? And that's why you surface sterilize and excise tissue deep down. You're trying to reduce the chance of just getting a fungus or a bacteria that's along for the ride. You're trying to get the real culprit. But there's primary and secondary infections. I could get a fungus that causes the initial disease. In comes a bacteria in the wound and takes over. The real problem wasn't the bacteria were there. The real problem was the fungus opening the door to let those bacteria in. So again, you have to demonstrate pathogenicity by isolating the suspected pathogen, growing up some of it, and then putting this cultured pathogen onto healthy plants and see if they become infected. Luckily, there's no people for the ethical treatment of plants, because now you're taking nice, happy, healthy plants and giving them a disease. But that's closing the circle. You have now demonstrated beyond any reasonable doubt that the organism that you identified as causing the original disease really is the correct organism, because you were able to create the same disease on other healthy plants using the material that you isolated. So, do you always have to do that? No. With experience, a good pathologist can sometimes do only parts of this and know exactly what they've got. They don't need to take it all the way to a level of scientific study that they're going to be able to publish a paper on it. And as a grower, you're not publishing a paper every time you get downy mildew or Phytophthora. You just want to know what you've got and move on. So you do what you need to do to get the information so you are confident that you have a plan to deal with it and move on. But if you're doing a true research, or you really have an unknown on your hands and you really are trying to say, is this really the right one? You've got to go all the way to the end and demonstrate pathogenicity. So let's take another quick break.